I think, you know, how it all started was actually just really small. So when we came back after Christmas holiday, was the time where it started out in Wuhan and in China. And I guess like for us, we just like looked at the situation, how it evolved there, and kind of like wrote a little article or blog post online where we said, like, you know, if it would actually swap over to Europe or to the UK, if we actually would be prepared in that case.
When we were talking with people here, we realized that a lot of the equipment required was already present. And a couple of people on site are already sort of experts in doing qPCR.
So right now we're trying to use OpenCell as a way to do a large-scale testing so that we can find and isolate the infections as quickly as possible.
We used our network to have a big open pool for anyone who might be able to help us. And quite quickly we had a team of over 200 volunteers.
So we were contacted as one of the candidates to build the containers for the laboratories to go inside of. We build stuff in containers all the time. We know how they work, they're modular, they can be transported.
So those are 4 of our mobile COVID testing laboratories. All the walls are hygienic wall cladding, hygienic wall flooring, and how it is actually a linear workflow. So you always walk in here and then you have to go outside at the other end.
This lab has been built absolutely specifically for the requirement of COVID-19. So everything is put exactly where you need it.
I think people get very excited about all of these novel technologies about coronavirus testing. What we're really doing here is already the gold standard test that laboratories around the world are already doing. And what was remarkable is that we were able to package it into one unit, one service, something that's much simpler.
The whole process is sort of separated into 3 separate steps. And so what happens in the first part is you have to take that sample, inactivate it, and sort of rip it apart. It's called lysis. You break apart the human cells, you break apart the viral particles, and you do that by adding chemicals to it. In station B, this is where essentially we're doing the technical processes called RNA extraction. And in the RNA extraction process, we essentially isolate that RNA and we wash it several times. So this is where the actual robotics comes into play, because this process is quite labor intensive. Essentially it involves binding that RNA using some sort of material. So in our specific case, we use something called a magnetic bead that's coated in something that will bind RNA, and then sticking that to a magnet and essentially washing it several times. And then the final step, station C, involves actually preparing the sample for PCR. So in this specific situation, for a coronavirus test, you do a certain type of PCR called reverse transcriptase quantitative PCR. Essentially what that does is take this viral RNA, convert it into DNA, and then essentially amplify it. Because at the end of this process, you may actually have very little viral RNA left. You also have to replicate it multiple times.
The automated process that we have means that in a single laboratory in a shipping container, we can get about 100 tests through every 45 minutes, running in parallel.
So we shipped our first COVID testing lab last week, and the entire OpenCell team is here in St. Heli at the moment. We start testing everybody that comes onto Jersey.
So at the current time, the tests are done during the day at the harbour, the airport, and up here at the drive-through.
Depending on the numbers that we have coming through today, is an exceptionally busy day. So we have over 400 passengers coming through today.
And the most important thing in this science is that we have no false positives and we have no false negatives, because that can cause chaos.
My name is Joe Prieto. I have done 2 years deployment in Afghanistan, where I was the lead microbiologist. The challenges will be at times very similar to those that we encountered in Afghanistan. The integrity of the samples will be checked, make sure that there are no leaking samples, that there is no contamination.
So it is a whole concatenation of actually quite complex setups in order to produce a good, reliable testing system.
Yeah, making this project open source and having it available for anyone in any country to replicate as quickly as they want to means that hopefully we'll be better prepared in future years for this crisis or similar crises.
The open source approach emphasizes reproducibility. It emphasizes something that can be collated and copied and then distributed.